I'm teaching a life science course in a large classroom format to graduate students and professional school students, like medical students and dental students. Some of the students asked me a question. How do I, as an instructor and a course director, read a textbook of the course? That was an interesting question, and I answered that I have experienced 4 phases of reading textbooks in my career so far. After hearing my comments, those students said that they had never heard of a viewpoint like mine before, and they found it interesting. So I would like to talk about my viewpoint today. This is related to how textbooks are different from primary research papers. I hope you will find it interesting too. Hi everyone, welcome to the Foundational Skills in Life Sciences podcast. I'm your host, Dr. Synaptologica. We discuss the skills that life science scholars learn, use, and improve throughout professional activities. Your benefit is expressed in my motto, learn the skills, enjoy academic life. Today's episode is the first one this year. So, Happy New Year. Let's have a fruitful year 2025. Today's episode. I serve as an instructor and as a course director of lecture series for graduate students and professional school students. When I chatted with those students, some of them asked about how I would read textbooks used in the courses. I answered them by giving practical details of how I would read textbooks. But at the same time, I explained my experience so far. The way I read textbooks has changed during the long course of my career. The students were very interested in those comments. So today, I would like to talk about how I read life science textbooks has changed and evolved over many years. In much later episodes, I would be happy to talk about the practical details of how to read textbooks. Just to give you a context, let me talk about 5 pieces of information about one course that I'm teaching and the textbook that we use there. 1. This is a life science subject. I studied it a long time ago when I was a student myself, but most of the subject is now outside my field of expertise. I do not need to mention the specific name of the course here because the name is not important, but consider biology, a large course that covers a very wide range of contents. 2. The course is of a large class format. It is a class of about 100 students or more. 3. For teaching this course, I have read one textbook thoroughly. And I'm also checking 2 more textbooks for comparing the contents and for learning further from them. 4. I read textbooks not only because I'm a good teacher, yes, I'm a good teacher, but also because I'm a course director. That means that I manage multiple instructors of the course. Together with the other instructors, I will think about how to run the course, what content is to be covered, and what exam questions to ask, etc. And 5, the textbooks are about 1,000 pages long, so they are thick and rich with information. These are the 5 pieces of information about one course that I'm teaching and the textbook that we use there. When I look back at how I read textbooks during my career, I can group my textbook reading years into 4 phases. I will talk about the first 2 phases today, and I will talk about the remaining 2 phases in the next episode. The first phase of reading textbooks. In my first phase, I was a student. I did not know how to read textbooks, and I did not even think that I would go through multiple phases in the future. What I did at the time was plain and simple. I tried reading the textbook from the first page without thinking much about how to read textbooks or why it is important to read textbooks. I tried understanding the content, and then I tried memorizing it. The focus was on memorizing the content. I think it is a standard way of reading as a student. I'm sure I was doing the same thing as my classmates were doing. I was in this phase throughout my undergraduate years and professional school years. My current students in my course would be in this phase too. Of course, it is possible that they are not reading any textbooks. They could only be focused on reading the lecture slides. Anyway, the second phase of reading textbooks. In my second phase, I stopped reading textbooks completely. So, no reading of textbooks in my second phase. I was in this phase throughout my postdoctoral years after graduation from graduate school. And this second phase lasted well after I became independent as a university professor. It ended only when I started to teach life science courses in a large classroom format. During this second phase, I focused on research. I worked with my lab members. I trained them, and I taught them the knowledge necessary for doing experiments and analyzing the experimental results. I read papers, I wrote papers, and I wrote grant proposals. I gave research seminars in the research communities inside and outside my university. I also gave some lectures in neuroscience courses. It is important to note that during the second phase of my textbook reading, all the activities were within my expertise. My input activities and output activities were limited to what I knew and what I could think about in my specific subfield in neuroscience. This makes a clear contrast with the first phase when I was a student. In the first phase, I did not have any expertise in any field. The second phase also makes a clear contrast with the third and the fourth phases when I worked outside my expertise. I will talk about the third and the fourth phases in the next episode. There are reasons why I did not read textbooks during my second phase. Now I'm going to talk about the 5 reasons. The second phase, reason 1. The first reason is simple. The textbooks were not necessary for me. All of my coursework had been finished, and I was not a student anymore in this phase, so there was no need for me to read course textbooks. From here till the end of today's episode, I'm going to talk about the remaining 4 reasons. They are more in-depth than whether I took a lecture course or not. They can be summarized into a single sentence in retrospect. Textbooks were not helpful, at least for me. A brief comment here. As I just said, I'm going to tell you 4 reasons, the reasons 2 through 5, about why I stopped reading textbooks in my second phase. Please let me make one comment first. Those reasons are related to how living in scientific research is different from learning from conventional textbooks. Those differences could sound like I'm criticizing textbooks, but I will not criticize textbooks. Instead, my goal is to say 2 things. 1, most of the textbooks are simply different from primary research papers. And 2, the textbooks have their own special, beneficial purposes. It is only that the textbooks that I read at the time were not helpful for me while I was focusing on performing primary research. As I will tell you in the next episode, textbooks will be important, especially for purposes other than primary research. Okay, let's move on. The second phase, reason 2. The second reason why I stopped reading textbooks in the second phase was that the textbooks provided information that was a little old. A major goal of most textbooks would be to summarize the extensive knowledge accumulated in wide areas of the field over long periods of time, like over decades to a century, depending on the fields, and to summarize it in an easily understandable manner. And such knowledge was contributed to by extensive activities of many scientists. Such knowledge was reported in the form of primary research papers, or sometimes in the form of books in the old days. textbooks tend to be a little old because the contents of most of the textbooks will be based on reviews of many review papers in the field. And the review papers will typically summarize the well-established results reported in the past. And establishing results can take time. So naturally, the contents of most textbooks tend to be old. In contrast, in scientific research, we need to keep our working knowledge in the field very up to date. The latest knowledge will be published in primary research papers these days. And we, the researchers, will read the primary research papers and judge them, learn from them, or discard them, even before they are discussed in review papers. So the second reason why I stopped reading textbooks in the second phase was that the textbooks provided information that was a little old. The second phase, reason 3. The third reason why I stopped reading textbooks in the second phase was that the textbooks provided information that was too simplified. The descriptions in most textbooks tended to be simple. Even complex phenomena with multiple variations are often described in a simplified manner so that the complex phenomena appear to be operating under just a single principle without any variations. It is reasonable for most textbooks not to deal with complex variations. Otherwise, the authors will need to include detailed variations of any complex phenomena, and such phenomena will appear in multiple wide fields covered by the textbooks. Then some readers could become confused because the content does not read simple. Some readers could also complain that the descriptions are too long or descriptions are not straightforward. So it is reasonable for most textbooks to be simple. But this oversimplification is a little risky if the textbooks serve as the main literature source for primary research. Let me talk about an example in neuroscience very briefly. Nerve cells, or neurons, in the brain communicate with each other in multiple different ways. In one conventional way, one neuron will send information to another neuron in one direction at the structure called synapses. Just to let you know, the study of synapses is synaptology, and it is the source of my name, Synaptologica. The efficiency of information transmission can be increased under certain conditions. But nerve cells in multiple brain regions show different degrees of changes, and the cellular and molecular mechanisms of how this efficiency increases seem different in different brain regions. Even in the same brain region, the mechanisms seem different at different types of synapses. So from a viewpoint of primary research, it is very dangerous to conclude that a single mechanism is working in all brain regions. If a single mechanism is described as if it is the widely accepted mechanism, or the dominant mechanism, or even the only mechanism, it could give a wrong impression that the single mechanism would be operating all the time and everywhere in the brain. It will cause confusion among researchers, and in a worst-case scenario, it could hamper the development of research. In fact, the primary research will often reveal variable phenomena or variable mechanisms in different brain regions, in different types of neurons, at different time points, and under different physiological and pathological conditions. Such a variety is excellent because it is a rich source of deep insight into reality. In my view, we would like to avoid oversimplification for research purposes. Now, variable phenomena or variable mechanisms would make it difficult for textbook authors to conclude in a simplified manner. Most textbooks had better be simple. So, textbooks provided information that was too simplified, at least for me. This was the third reason why I stopped reading textbooks in the second phase. The second phase, reason 4. The fourth reason why I stopped reading textbooks in the second phase was that the textbooks tend to avoid the current controversial topics. If textbook authors want to provide well-established information of the past, especially in a simplified and concise manner, it is reasonable that the textbook authors would want to avoid controversial topics. By definition, controversial topics have not been settled or resolved yet at the time of writing. That means that there is no clear-cut answer to the questions in the topic. The reality of science at the time shows at least two possible scenarios or explanations, and so the situation remains unclear. Because of unclear statements, the writing could appear confusing to some naive readers, and such readers might complain that the descriptions are not good in the textbooks. Also, the textbook will become longer if such controversial topics are included in many of the covered fields. Some readers could complain that the descriptions are unnecessarily long, and also the textbooks are limited in space usually. Good textbook authors will listen to the feedback from readers. If the feedback is negative, based on the features that we just talked about, the authors could edit out the current controversial topics. In contrast, in research, the current controversial topics are very juicy. The word juicy might be a little informal. We would say that the current controversial topics are intriguing, fascinating, and captivating. In other words, they are interesting and they can be good targets of research. So, some researchers would want to pay attention to controversial, hot topics that are ongoing now, and they would pick them up and try deriving clear answers to the issues. So the fourth reason why I stopped reading textbooks in the second phase was that the textbooks tend to avoid the current controversial topics. The second phase, reason 5. The fifth and the last reason why I stopped reading textbooks in the second phase was that the textbooks might not be optimal for sharpening our thinking processes for primary research. When we start training for primary research, one key thing is to learn how to think. We want to learn at least two core features of how we think. They're about how to develop and refine new concepts and new methods. By these developments and refinements, I mean the following. For the concepts, we want to learn how the researchers came up with the original ideas, how the new ideas were different from the old ones, how the new ideas were tested, and how the new ideas were further modified, and so on. For the methods, we want to learn how the new experimental methods were different from the old ones, advantages, disadvantages, and limitations of the methods, changes in the background knowledge, how to design experiments based on the available methods, how to do the experiments, how to analyze the experimental results, and so on. We can learn these developments and refinements in concepts and methods most effectively by studying the original publications of primary research. And I read many primary research papers. I also read books if the original ideas or methods were historically important and if they were described in books. So the fifth reason why I stopped reading textbooks in the second phase was that the textbooks might not be optimal for sharpening our thinking processes for primary research. This is the end of the second phase of my reading textbooks, or not reading textbooks. Summary. What is a lesson today? Today I talked about the first 2 out of the 4 phases of how I read life science textbooks so far. In the first phase, I was a student. I simply read textbooks without thinking much about how to read textbooks or why it is important to read textbooks. And I tried remembering the contents. In the second phase, I focused on primary research. During this phase, I did not read textbooks. It was because the textbooks were not necessary for me without taking courses, and because the textbooks were not helpful, at least for me, as a junior researcher. I was not clearly aware of these reasons when I was in the first and the second phases, but I can state the reasons clearly now in retrospect. Today's lesson will be this. You do not have to follow the phases that I experienced. Your experience will be different from mine. But it will be helpful for you to know that there are differences between textbooks and the primary research papers. They are different in the contents. Textbooks cover well-established knowledge in wide, related fields, whereas primary papers are focused on a narrow question set in a narrow field. Textbooks and primary research papers are different in the lengths and the styles too. And so, textbooks and primary research papers will serve different purposes. Knowing these purposes will help us appreciate the different roles that they play in our academic lives. We will talk about the roles of textbooks in the next episode. This is it for today. In the next episode, I will talk about the remaining 2 phases of my experience of reading textbooks. I have evolved after my first and the second phases. How have I changed? Please wait for the next episode. Your action. Please tell me how you're reading textbooks. If you're not reading them, please tell me how you read them before and why you're not reading them now. You can teach me at my email address, ideas@synaptologica.com. We can think deeply by sharing our experiences together. Thank you for joining me in today's episode of Foundational Skills in Life Sciences. I'm Dr. Synaptologica. I'll see you soon. Bye for now.